Welcome to another IB Environmental Systems and Societies video. Today we're looking at ESS topic 2.5, Investigating Ecosystems. This section of the ESS course focuses on the practical methods you're going to use to study ecosystems. Understanding these techniques isn't just about passing your exams. These are the same tools that scientists use worldwide to monitor environmental change and develop conservation strategies. They're also really useful for your internal investigation. So we're going to explore how to quantify, analyze, and understand the natural world. Let's get into it. The first significant idea here is fundamental. Ecosystem investigations allow us to make comparisons between different ecosystems and track changes over time. Think about why this matters. Without standardized methods to study wetlands like the one shown, we can't determine if habitats are improving or degrading, and we can't understand how human impacts are changing natural systems around the world. Our second significant idea emphasizes quantification. Nature doesn't arrange itself neatly for our convenience, so we impose measurement systems on it. When you count plants or measure soil pH, you're converting the messy complexity of nature into data that can be analyzed. This transformation from qualitative observation, that's observation that you describe with words, to quantitative data, that's numerical data, is what allows us to draw meaningful conclusions about environmental patterns and processes and human impacts on them. Every ecosystem investigation has to start with proper identification. Just as you have a name and address, ecosystems need precise names and locations too. This allows other scientists to find your study site, to replicate or repeat your work, or track changes over time. The specificity matters. Saying a forest in Switzerland is really different from identifying Dinkerwald, a mixed deciduous coniferous managed woodland in Bar, Switzerland. The second one is a lot more precise and allows other people to find and repeat your work. Organism identification is essential for ecological studies. Dichotomous keys are brilliantly simple tools. Each step presents just two options, gradually narrowing possibilities until you identify your specimen. Those options are generally a yes or a no question. Remember that effective keys use clear comparative characteristics rather than subjective terms. A good key might ask, does the leaf have a serrated edge? Rather than, is the leaf pretty? This more complex dichotomous key for river macroinvertebrates shows how keys work in practice. Notice how the structure branches repeatedly, with each decision point narrowing the possibilities. This systematic approach transforms identification from guesswork to science, and that allows even non-specialists to accurately identify species. This is an important skill for ecosystem monitoring and assessment. Sampling strategies allow us to study large areas by examining representative parts of that area. This slide shows how we might measure changes in space or along gradients over time, through succession, or before and after human impacts. Quadrats are among our most fundamental tools for environmental monitoring. These simple frames define precise areas where we can count organisms or measure environmental factors. Transects are another essential sampling technique, and they allow us to study how conditions change across environmental gradients. The point transect shown here records data at specific points along a line. This method is perfect for studying zonation, how species distribution changes with distance from a shore, up and down an elevation on a mountain, or along other gradual environmental transitions. Belt transects combine benefits of quadrats and point transects. By placing quadrats at intervals along a transect line, you can get detailed information at regular distances. This technique excels at capturing how community composition shifts or changes across boundaries like forest edges, shorelines, or pollution gradients. This is precisely the kind of transition that often interests environmental scientists the most. How we arrange our samples dramatically affects our results. Random sampling prevents bias, but it might miss important patterns. Systematic sampling ensures even coverage, but could coincide with environmental patterns. Stratified sampling targets different habitat types deliberately. Each one of these approaches has strengths and limitations, and your research question should determine which method you choose. Here's a real quadrat in action, showing how this simple square frame creates a defined study area. Notice how it captures a small, manageable section of the ecosystem that can be thoroughly inventoried. The power of quadrats comes from their repeatability. By using the same method in the same way in different locations or times, you can make valid comparisons across space and time. Scientific conclusions are only as good as the data they're based on. Repeating measurements increases reliability by reducing the impact of random errors or anomalies. The number of repetitions needed varies by factor. Highly variable measurements like species counts might need many repetitions, while stable factors like soil type might need fewer repetitions. Measuring biomass helps us understand energy flow through ecosystems. The process requires careful sampling, drying to remove water content, and extrapolation to estimate total biomass. Notice the challenge mentioned. What about root biomass? This highlights how invisible components of ecosystems are frequently overlooked, but they may represent pretty significant portions of the total biomass and ecosystem function. Organisms can either be fixed in place, like vegetation, or they can move around. We call those organisms that move around motile organisms. In the US, a lot of times you'll see mobile organisms. For non-motile organisms like plants, we have several measurement techniques. Actual counts work for larger, distinct individuals, like trees. Percentage cover estimates the proportion of area covered by a species. That's useful for grasses and mosses. Percentage frequency records how often a species appears in sample plots. Each method suits different research questions and the type of organism. Estimating populations of moving organisms presents unique challenges. Direct methods like visual counting can be accurate, but that's often labor intensive and it's impractical for large areas or elusive species that are hard to find. The tagged dragonfly illustrates how scientists directly mark individuals for later identification. That's part of a more sophisticated population estimation technique. The Lincoln Index provides an elegant solution for estimating wildlife populations. By capturing, marking, and recapturing organisms, we can calculate total population size without counting every individual. This is the method that you should remember for estimating population size. Capture, mark, recapture is called the Lincoln Index. The formula looks complex, but it follows simple logic. The proportion of marked animals in the second sample should reflect the proportion of marked animals in the whole population. For your exams, you don't need to memorize the formula because it will be given to you, but you absolutely must know how to use and apply it. Species richness is simply the count of different species present. That's a fundamental biodiversity measure. The illustration contrasts two communities with identical species richness, but they have different species evenness. Community 1 has balanced populations, while Community 2 is dominated by a single species. This shows why richness alone doesn't tell the complete story of biodiversity. Species diversity indices can indeed feel overwhelming. The key point is that diversity combines both richness, the number of species, and evenness, the relative abundance of those species. Think of diversity indices as mathematical tools that compress complex community data into single, comparable numbers. That's important for monitoring changes over time. This visual perfectly illustrates the difference between species richness and species evenness. Both communities contain 4 tree species, but Community 1 has equal numbers of each, that's high evenness, while Community 2 is dominated by yellow trees. That means that Community 2 has low evenness. This distinction matters ecologically. Diverse, even communities usually show greater stability and resilience to disturbance. The Simpson Diversity Index formula calculates diversity based on both richness and evenness. Higher index values indicate greater diversity. Remember that this index is most valuable for comparing similar habitats or tracking changes in one location over time. The formula may seem intimidating, and you don't need to memorize it for the exam because, like the Lincoln Index, you'll be given the formula. But calculators and spreadsheets can make application pretty straightforward, and the numbers you'd be given on your ESS exam will be simple enough that you can do it in just a few minutes. The most important thing is that you know how to use the Simpson Index and when to use it. Ecological investigations form the perfect basis for your ESS internal assessment, or IA. Notice how the components align perfectly with IA requirements, identifying the context, planning your investigation, analyzing your results, evaluating the methods that you used, applying your findings, and of course, clear communication using scientific terminology. A well-designed ecosystem investigation shows your environmental understanding while developing transferable research skills that you can use elsewhere. Creating identification keys is a practical skill you should practice. It will help you understand how to use a dichotomous key if you see one on your exam. When making keys, focus on obvious, permanent features that clearly differentiate species. Avoid subjective descriptions like beautiful or large in favor of measurable characteristics like leaves over 10 centimeters long or stems with visible thorns. Critical evaluation of sampling strategies demonstrates higher level thinking. Consider how random sampling prevents bias, but it might also miss rare species, while systematic sampling ensures coverage of the entire area, but it might coincide with environmental patterns. The best strategy depends on your research question, the available resources, and the ecosystem being studied. These TOK connections highlight important questions about the method, validity, and scope of investigations. When is quantitative or numerical data superior to qualitative descriptive observations? How does knowledge gained through field observation compared to controlled lab experiments? These kind of questions invite us to consider how we know what we claim to know about environmental systems. That's it for ESS Topic 2.5, Investigating Ecosystems. You should be able to describe these ecosystem investigation techniques and when to apply them for success on your exams. These are the same methods used by scientists worldwide to monitor environmental change, to develop conservation strategies, and to understand our changing planet. Good luck, and until next time, happy learning.